What if you could press pause on life? Not just for a moment, but for months or even years, waiting out the harshest winters or the driest droughts, only to resume exactly where you left off when conditions are perfect again. This isn't science fiction, it's a biological reality for hundreds of thousands of species, from tiny insects to massive grizzly bears. It's called developmental diapause, a state of suspended animation that is far more complex and fascinating than simple sleep. Today, on G Fun Facts Online, we're unraveling the molecular symphony that allows life to hit the pause button. Diapause isn't just about taking a nap when things get tough. That's called quiescence, a simple, direct response to a bad situation, like a bug hiding under a rock when it gets too hot. Diapause is a proactive, genetically programmed strategy. Organisms that use it anticipate predictable hard times, like the coming of winter, and shut down their development in advance. This programmed arrest can happen at almost any life stage. The silkworm, for example, enters diapause as a tiny embryo inside its egg. In contrast, the monarch butterfly undergoes reproductive diapause as a full-grown adult. This state of suspended fertility is what allows it to undertake its epic, multi-generational migration from Canada to Mexico without stopping to reproduce. The sheer diversity of creatures that use diapause, from insects and crustaceans to fish, and over 130 species of mammals, tells us that this strategy is a massive evolutionary advantage. It has evolved independently, time and time again, as a master key to survival. So how does an organism know when to hit the pause button? It listens to the rhythms of nature, using specific environmental cues. The most reliable signal, especially in temperate regions, is the changing length of the day, or photoperiod. As summer turns to autumn, the days get shorter. For an insect, this is a blaring alarm that winter is coming. They measure this change using their internal circadian clocks, the same molecular machinery that regulates daily rhythms of sleeping and waking. A popular theory, the external coincidence model, suggests the clock creates a specific window of time each day. If this window falls during the darkness of a long night, a signal is sent, induced diapause. If it falls during the light of a short night, the signal is, keep developing. The core of this clock is a network of genes with names like period and timeless, which create a daily molecular rhythm that translates day length into a developmental decision. Temperature is the other major conductor in this orchestra. It often works with day length to fine-tune the response. For some species, like a specific strain of silkworm, temperature is the main trigger. If they experience temperatures above 20 degrees Celsius during development, their offspring are automatically programmed to enter diapause. The sensation of heat and cold involves specialized molecular thermometers, like the TRPA1 protein channel, which sends temperature information to the nervous system. Once these environmental cues are detected, they need to be translated into a body-wide response. This is the job of the neuroendocrine system. Think of the brain as the orchestra conductor. It integrates the information from day length and temperature and relays commands to a series of glands. In insects, these glands produce two superstar hormones that regulate almost everything: juvenile hormone, or JH, and ecdysone. The changing levels of these hormones are what ultimately orchestrates the entire diapause state. Let's zoom in on the molecular soloists in this symphony, because this is where things get really fascinating. A central, ancient, and incredibly important pathway that controls diapause is the insulin signaling pathway. You've probably heard of insulin in the context of human metabolism and diabetes, and its role here is related. This pathway is a master regulator of growth, metabolism, and aging. During diapause, this pathway is shut down. In the mosquito Culex pipiens, the species that pesters you on summer evenings, short autumn days cause the insulin signaling pathway to go quiet. This is the critical first step for inducing reproductive diapause, allowing the female mosquito to survive the winter. When the insulin pathway is shut off, it unleashes a powerful transcription factor named FOXO. Normally, insulin signaling keeps FOXO locked up in the cell's cytoplasm, but without that signal, FOXO moves into the nucleus, where it can access the DNA. Once there, it acts like a general, turning on a whole suite of genes responsible for stress resistance, longevity, and crucially, building up fat reserves. This activation of FOXO is what transforms the mosquito into a winter-ready survivalist. The fact that this same insulin FOXO system controls dormancy in fruit flies, and even the nematode worm C. elegans tells us it's an ancient molecular tool for hitting pause. Other hormones play more specialized roles. Juvenile hormone, or JH, is the key conductor of reproductive arrest in adults. In many insects, like the lady beetle, entering diapause requires a steep drop in JH levels. This shuts down the ovaries and halts all reproductive activity. Conversely, applying a dose of JH can often wake an insect up from its adult diapause. Then, there are the EC dysteroids, the molting hormones. A halt in development during the larval or pupal stage is often caused by a drop in ecdysteroid levels. The brain simply stops sending the signal to produce them, and development grinds to a halt. It's like a factory shutting down the assembly line by cutting power to the main conveyor belt. But it's not just about hormones. To maintain this state of suspended animation for months, an organism needs to make profound, stable changes to its gene expression. This is where epigenetics comes in. Epigenetics refers to modifications that change how genes are used without altering the DNA sequence itself. Think of it like putting sticky notes on a musical score. The notes are all still there, but the sticky notes tell the orchestra which parts to play loudly, which to play softly, and which to skip entirely. One way to do this is through histone modification. Histones are the proteins that DNA wraps around. By adding or removing chemical tags to these histones, you can either pack the DNA up tightly, making genes inaccessible, or open it up for reading. During diapause in fruit flies, for example, certain active histone tags are removed in the ovaries, helping to cause the global shutdown of gene expression needed for reproductive arrest. Another powerful epigenetic tool is microRNAs. These are tiny molecules of RNA that don't code for proteins themselves. Instead, they act as molecular silencers. They find specific messenger RNA molecules, the blueprints for proteins, and either destroy them or block them from being used. In diapausing insects and worms, specific microRNAs are switched on to silence genes that promote development, while others are switched off to allow the expression of genes that enhance survival and stress resistance. They are the tiny, precise conductors of this grand genetic symphony. With all these systems in place, what actually happens inside the cells? First, there's a dramatic metabolic suppression. The organism's entire metabolism slows to a crawl. Oxygen consumption can drop to just a tiny fraction of normal levels. This is essential for conserving energy. You can't be burning through your food reserves if you have to survive for 6 months without eating. The body switches from burning carbohydrates to relying on lipids, or fats, which are a much more efficient long-term energy store. To survive freezing temperatures, many insects also produce cryoprotectants, natural antifreezes like glycerol and sorbitol. These chemicals prevent the formation of sharp, cell-destroying ice crystals. The second key cellular event is cell cycle arrest. Development is, at its core, cell division. To stop development, you have to stop cells from dividing. During diapause, molecular brakes are slammed on the cell cycle. Key proteins that drive cell division, like cyclins, are downregulated, while inhibitory proteins are activated, locking cells in a non-dividing state until the signal comes to wake up. And this incredible strategy isn't just for insects. Many crustaceans, like the brine shrimp Artemia, produce diapausing eggs that are legendary for their toughness. These sea monkeys can survive for years in a completely dried-out, oxygen-free state, only to hatch within hours of being placed back in salt water. They use many of the same tools, including the insulin pathway and a large suite of protective chaperone proteins that prevent other cellular machinery from degrading. Then there are the mammals. Over 130 species, including bears, seals, badgers, and some mice, use embryonic diapause. The mother's body puts the development of a newly fertilized embryo on hold. The tiny ball of cells, the blastocyst, just floats in the uterus, waiting. This allows the mother to time the birth of her offspring perfectly, with when food will be plentiful and conditions are favorable. In mammals, this process is controlled by maternal hormones like progesterone and estrogen, and a key cell growth pathway called mTOR is suppressed in the embryo to maintain the paused state. Eventually, the pause must end. The termination of diapause is triggered by another set of environmental cues, like a long period of cold followed by warmth, or the return of long days in spring. These signals reactivate the whole system. The brain releases hormones, ecdysteroid levels surge in a pupa, and the cell cycle breaks are released. In the silkworm embryo, waking up is linked to a specific signaling pathway that senses temperature changes, triggering the reconversion of stored antifreeze back into usable energy and kickstarting development. The study of diapause is more than just a fascinating glimpse into the resilience of life. It has huge practical implications. Understanding how to manipulate diapause could give us new ways to control insect pests, perhaps by preventing them from entering their winter slumber, leaving them vulnerable to the cold. For beneficial insects like commercial silkworms or the bugs we use for biological pest control, being able to turn diapause on and off on demand would be incredibly valuable. But the insights go even deeper. Diapause is a masterclass in how to manage aging and resist stress. And astonishingly, scientists have discovered that some cancer cells can enter a diapause-like state to survive chemotherapy, hiding out in a dormant phase only to re-emerge later and cause a relapse. By understanding the molecular tricks of diapause, we might learn how to target these dormant cancer cells. The possibilities are profound, from preserving organs for transplantation to potentially discovering ways to extend human health span. Developmental diapause, once a biological mystery, is now revealing itself as an evolutionary masterpiece, a testament to life's intricate and elegant ability to wait for its moment in the sun